Hello and welcome to some biology class. Friends, today we shall discuss about ABC transporters. The ATP binding cassette transporter, also known as ABC transporter superfamily, contains membrane proteins that transport a wide variety of substrates across extracellular and intracellular membranes, including sugars, amino acids, heavy metal ions, conjugates, peptides, lipids, polysaccharides, xenobiotics and drugs. The transport is ATP dependent. Overexpression of certain ABC transporters occur in cancer cell lines and tumors that are multi-drug resistant. Genetic variation in ABC genes is the cause and contributor to a wide variety of human disorders with Mendelian and complex inheritance, including cystic fibrosis, neurological disease, retinal degeneration, cholesterol and bile transport defects, anemia, and drug response phenotype. Studies on ABC transporters began in the early 1970s. They are present in all living organisms. In addition to transport, they are also involved in biochemical and physiological processes. In human beings, ABC transporters are known to function as ion channels and receptors. Some of the ABC transporters function as importers, which have been confirmed in prokaryotic organisms. Many ABC transporters act as exporters. The structure of ABC transporter is highly conserved among most eukaryotic organisms. A functional ABC transporters consist of two transmembrane domains and two cytosolic nucleotide binding domain. All of them are combined in a single polypeptide chain, whereas a half transporter contains one transmembrane domain and one cytosolic nucleotide binding domain. They are sometimes encoded as separate polypeptide which are later fused to form the functional transporter. This figure shows the structure of a typical ABC full transporter. The transporter has two transmembrane domains and two nucleotide binding domains. This is the lipid bilayer, and here this is a transmembrane domain and that is another transmembrane domain. Each transmembrane domain has 6 alpha helices. The nucleotide binding domains are cytoplasmic, and this is the carboxyl terminus and this is the amino terminus. So in a ABC full transporters, two transmembrane domains and two nucleotide binding domains are combined in a single polypeptide chain. The nucleotide binding domain of ABC transporters contain several highly conserved nucleotide binding sequences which are Walker A and B motifs separated by 90 to 120 amino acids. It also has a D loop that has an aspartate residue. Another sequence is a Q loop that has a glutamate residue. The NBD also has a H motif that has an invariant histidine active site. In addition to this, the nucleotide binding domain has an alpha helical region which has the ABC signature sequence. All these sequences are combined in the nucleotide binding domain of ABC transport. This figure shows the most common arrangement of a typical ABC full transporter, in which this is the amino terminus, after which the first transmembrane domain is present. The first transmembrane domain is followed by first nucleotide binding domain, after which the second transmembrane domain is present. After second transmembrane domain, the second nucleotide binding domain is present, that is followed by the carboxyl terminus. In addition, other types of arrangement as shown in second, third and fourth are also seen in some cases. Phylogenetic analysis places the 48 known human ABC transporters into 7 distinct subfamilies. Their precise map location on human genome, their expression data and their localization within the ABC superfamily have already been determined. ABC pumps are mostly unidirectional. In bacteria, ABC transporters are predominantly involved in the import of essential compounds that cannot be taken up into the cell by the process of diffusion. In eukaryotic organisms, ABC transporters are mostly involved in the movement of compounds from cytoplasm to the outside or into the intracellular organelles such as mitochondria, endoplasmic reticulum and peroxisome. Most of the known functions of ABC transporter in eukaryotic organisms is the shuttling of hydrophobic compounds within the cell or outside the cell. Among various models that have been proposed to explain the mechanism of transport by ABC transporters, ATP switch model provide a reasonable framework for transport mechanism. According to ATP switch model, during transport, repeated communications occur between the transmembrane domain and the nucleotide binding domain in both directions and involves only non-covalent conformational changes. The transport process is initiated by binding of the substrate to the transmembrane domain and subsequent structural changes are transmitted to the nucleotide binding domain, which includes binding of ATP and formation of a closed dimer of nucleotide binding domain. The closed dimer of nucleotide binding domain induce substantial conformational change in the transmembrane domain. This conformational change initiates the translocation of the substrate through rotation of the transmembrane domain and opening towards the extracellular space. Finally, ATP is hydrolyzed and ADP and inorganic phosphate are released and the closed dimer is converted back into the open dimer to initiate a new round of transport cycle. So friends, this is all about structure and function of ABC transporters. For more updates, please subscribe Sam by class. Thank you.